Hi everyone, my name is Francesco Marangoni. I am a tumor immunology in the Department of Physiology and Biophysics. And this lesson will be about, indeed, the fundamentals of cancer immunology and immunotherapy. I am very sorry not to be able to present this to you in person. However, if you have any question about the presentation, please reach out to my email address, f.marangoni@uci.edu. So the objectives of this lecture will be, first of all, to refresh some fundamental concepts in tumor immunology and immunotherapy. Then we will look at several cell types and how we can actually turn them into drugs to fight tumors. The first will be T cells, and among the T cells, while we speak about T cells, we'll also revisit the principles of checkpoint blockade immunotherapy. Then we will look at B cells and the use of antibodies as drugs in oncology. And we also discuss a new strategy, which is basically to put together characteristics of both B and T cells to devise the CAR T cell therapy. Finally, we will discuss some aspects of the innate immunity to tumors and how this can be leveraged against cancer. Let's start from the very fundamentals. So, up to the 70s, the tumor was really thought or conceptualized as a cell autonomous disease. So in following several types of insult, cells in our body will suffer a DNA damage. And in some instances, this DNA damage is actually targeted or involving genes that control cell death and cell growth. When this happens, and when the number and quality of insult is high enough, the cells basically go crazy and start proliferating without any control, giving rise, of course, to a neoplastic mass. So this is a vision that basically indicates the culprit is exclusively the tumor cell. The tumor grows because the tumor cells never stop proliferating. However, more recently, we basically assisted to a complete paradigm switch. So the tumor is not only growing because of the tumor cells, but also because of normal cells that are part of the tumor and that help its growth. Let's think about blood vessels, for instance, providing food to the tumor. Let's think about fibroblasts that provide the extracellular matrix, and several kinds of immune cells. Some of them will be able to fight the tumor, but some of them, unfortunately, favor tumor growth. So the tumor is not only growing because of neoplastic transformed cells, it is also growing because of the help of normal cells. And this opens up a whole new world of potential therapies. Indeed, the therapies we have for tumors essentially co-evolve with our understanding of the tumor itself. When we were sure in the 70s that the tumor was just composed of tumor cells, the therapies were basically targeted to either destroy the mass using surgery or radiotherapy, or they were aiming to stop proliferation, like for chemotherapy, which kills only proliferating cells. However, when we understood that the blood vessels are important for the tumor, and the concept of anti-angiogenic therapy became of age, so starting to, impair the angiogenesis, the tumoral angiogenesis, to basically prevent food from reaching the cancer. More recently, when we understood that some cell types actually go, some immune cell types go to the tumor, and those immune cell types may have an anti-tumor function if they are properly instructed, this gave rise to a lot of emphasis in instructing those cells in killing the cancer, or taking some of the characteristics of those cells and using those to kill cancer. Let's think about antibodies produced by B cells. Those were purified and used as anti-tumor drugs. Let's think about cytokines that drive T-cell attack. Let's think about using the T cells that are specific for antigen presented by the tumor, or CAR T cells. However, there is a fourth level of control, and which is the emphasis of the last, probably 10 years or so. The tumor is actually able to tweak the immune system, to trick the immune system, to actually enact or favor immunosuppressive mechanisms. And those immunosuppressive mechanisms basically dampen the anti-tumor immune response and favor immune growth. Let's think, for instance, about T regulatory cells. We'll talk about them quite a bit today. Also macrophages that in tumor are called TAM, as tumor-associated macrophages. And first and foremost, checkpoint blockade, that are molecular mechanisms that basically suppress the anti-tumor activity of T cells. So if we unleash those, if we remove the brakes, we'll get a much better anti-tumor immune response. So the first and principal aspect of tumor immunotherapy relies on how the body recognizes there is a cancer. So you may recall from basic immunology studies that we have evolved during the ages immune mechanisms that actually are targeted towards something that is recognized as non-self, so extreme, foreign to us. And at the same time, we have mechanisms to immunosuppress every response that is directed against something that is recognized as self, as something belonging to us. And this is extremely important because, of course, it is evolutionarily lame to allow the immune system to attack ourselves. Therefore, we have developed regulatory cells and other mechanisms that, in normal situation, block our immune system from reacting against us. However, the tumor is developing from our own cells. How is it recognized as something foreign? And the reason is that the same mutations that alter the DNA of neoplastic cells to dysregulate their proliferation and survival actually alter the expression and the characteristics of many other genes and generate new epitopes, new proteins, or in one word, neoantigens, that they are not perceived as self, therefore recognized by the immune system, especially by CD8 T cells, which may definitely start an anti-tumor immune response. And this is a list, not included in the final exam, just telling you an example of some of those tumor-associated antigens that have been identified over the years and for many tumor types. Note that every tumor type oftentimes has specific tumor-associated antigens. We'll get back to that and how this is basically leveraged for therapy. So, are all the tumors equally mutated or equally having neoantigens that arise from mutations? Absolutely not. They're totally heterogeneous in that. Some tumors, like this kind of astrocytoma, the pilocytic astrocytoma, is essentially non-mutated. Other tumors have different levels of mutations, with the tumors that are the most mutated, melanoma, for instance, a skin tumor, and lung cancers, several types of lung cancers. As you see here, they are heavily mutated. Why they are heavily mutated? Well, because they are, generally speaking, exposed to a lot of insults. The skin is exposed to sunbeam, and this is really the reason why we need sunscreen to prevent the accumulation of too many mutations in the skin. The lung, of course, is exposed to every kind of pollutants. Therefore, the tumors that are the most exposed to insults are probably also the ones that are going to be the most mutated and the most immunogenic. And this leads us to probably the most important immunological concept for today, which we essentially summarize how our immune system sees the cancer and how our immune system responds to the cancer, and how these two aspects, tumor recognition, tumor response, are intertwined, are constantly intertwined to basically create what is called here as a cancer immunity cycle. So let's start from point one. Point one is the death of tumor cells. So, every cell in the body has a natural lifespan. Every cell in the body, it eventually dies and releases the proteins that it contains. This is no exception for tumors, which when they die, they release antigens, including mutational neoantigens, in the tumor environment. Those antigens are picked up by specialized immune cells. They are called dendritic cells, which actually have a very unique, several unique characteristics, but one of them is to be present in cancer, gobble up antigens, including neoantigens, and then migrate from the tumor to the lymph node. The lymph node is the regional headquarters where the immune response is organized. While in the lymph nodes, these dendritic cells, and just I open a little, a little incidental sentence or incidental concept here. The dendritic cells are among the very few in the body to be able to retro-migrate, so to speak, from tumors to lymph nodes. Many other cells do only the opposite route. However, the dendritic cells homing to the node present the antigens that are recognized by naive T cells, and those naive T cells, when they recognize their cognate antigen, they are primed. So they see the antigen for the first time, proliferate like crazy, acquire cytotoxic characteristics, and redistribute to the whole body through the blood. So the blood is definitely a sort of highway in our body, but those cells that are primed and now flow in the blood are basically also equipped with several chemokine receptors that migrate the infiltration to inflamed areas, including the cancer. So when they get into a blood vessel that is basically feeding a tumor, they recognize that area is inflamed, and the cells actually transmigrate out of the blood vessel, out of the highway, and they start to migrate in the interstitium, which is basically, inflamed tissue that much likely is also neoplastic in this case. So when in the tumor, the cells recognize the antigens, the T cells recognize the antigen for the second time. And this second activation unleashes all the cytotoxic mechanisms that the cells have, leading to more tumor cell death. And this closes the cycle. This closes the cycle because we generate more neoantigens that are taken up by more dendritic cells, which migrate to the lymph node to prime more T cells, and so on and so forth, till the tumor basically disappears. So you may tell me, if we have such nice conceptualization of the crosstalk between tumor and the immune system, why in the world patients have tumors? Why in the world the tumor actually manages to escape the immune system? and kill the patient. Well, the reason of that is that the tumor, of course, enacts strategies to basically not be seen or to suppress the tumor immunity cycles. So we'll review some of them. The first three of them is how the tumor essentially tries not to be seen. So in other words, the tumor tries not to be killed by stealthing himself or itself from recognition of effector cells. In other words, the immune mechanisms that have the potential to get to the tumor and kill it, they never do, because the tumor is almost invisible to them. Three are listed here, and we'll review them in detail. However, the first two of those mechanisms are extremely similar to each other. So, for instance, a mutation in the tumor, for instance, the tumor may be extremely poorly mutated. We have seen the example of astrocytoma, or accumulate mutations that interfere with the ability of tumor cells to present antigens for recognition by T cells. So if there is no T cell tumor recognition, the T cells that come to the tumor and try to kill it, they never see the antigen the second time. This is the sixth step of the tumor immunity cycle, and the tumor cell will survive. So the second mechanism, escape, is tightly bound to the first, and it works more or less as follows. So that the lack of mutations or the lack of the genes for antigen presentation may be mediated not by, an interesting characteristic of the tumor stopping to produce them, but they may be secondary to actually immune pressure. In other words, it is a Darwinian system. The T cells come in, they do destroy the cells that present antigen nicely, or that have a lot of neoantigens. However, some of the tumor cells may be selected because they express either very little neoantigens or very little MHC molecules. And in that case, you have escape from an initial immune pressure. There is a third way a tumor can actually stealth himself from recognition, which is actually what is so-called cold tumors in the current, immunological language. This essentially is a defect of dendritic cells. So the dendritic cells, that is, they link the step 1, 2, up to 3 of the cancer immunity cycles. They don't do their job, and they don't do their job for a couple of reasons that are reported in literature. One of them is that they are not recruited to the tumor. And the second reason may be that even if they are able to go to the tumor, they are basically blocked in their ability of picking up antigen and starting the cancer immunity cycle. So in the figure here, you see a tumor that is basically devoid of any immune cells, which are represented in red, and this is considered a cold tumor. And the picture here at the bottom is, on the other hand, just for contrast, how a hot tumor looks like. So a tumor that has a lot of dendritic cells, and the cancer immunity cycle has started. Therefore, you may also see a lot of T cells there. So what are the effector mechanisms that the tumor may try not to elicit? Those are T cells, especially CD8s and CD4s, some kinds of dendritic cells, such as the DC ones. Of course, the tumor doesn't want to elicit an antibody response from B cells as well. And so it will also try not to have antibodies around. And the last mechanism is pro-inflammatory macrophages, tumor-associated macrophages. So nomenclature that I don't like because it is oversimplistic defines them as M1. The concept that we can retain for now for this lesson is that M1 macrophages are actually anti-tumoral. However, they are hardly ever present in cancer, and we will get back to that at the end of the lesson. The tumor, so we have just reviewed, they can escape immune recognition by blunting effector mechanisms. That's not the only mechanism of escape they have. The second class of strategies actually involve active immunosuppression. So the tumors, in this case, they are not trying to hide themselves from recognition. They are trying to avoid the deleterious effect of recognition. And this occurs through at least 3 different mechanisms. One is tolerance, which is extremely important for tumor immunotherapy because checkpoint blockade immunotherapy. Because tolerance is what happens to a T cell that initially recognizes and destroys tumor cells. However, upon chronic exposure to the same cognate antigen, the T cells becomes less and less functional. So over time, it becomes tolerant. And when you get to that level of tolerance, to high levels of tolerance, basically the T cells don't deploy anymore their cytotoxic arsenal to the tumor, and the result is that the tumor grows. So molecules that actually are important for that are nothing less than PD-1 and CTLA-4. Exactly the main blockers, the main brakes for T cell function in tumors that are, of course, the target of one of the most advanced therapies that we have today, which is tumor checkpoint immunotherapy. The second mechanism is for the tumor to recruit immune cells that are immunosuppressive, and 2 are the main cells doing that. One is T regulatory cells, which is T cell, that is totally, whose only job is to keep things calm, to switch an immune reaction off. Of course, they evolved to avoid autoimmunity, so an aggression of the immune system against us. But in the case of tumors, they actually do a very bad or unpleasant job, which is actually to switch off the anti-tumor immune response. So in cancer, T regulatory cells are one of the main providers of tumor immunosuppression. The other cell type is an anti-inflammatory, or if you want, alternatively activated macrophage, which the nomenclature I don't appreciate calls M2. Those cells are very abundant in tumors, and unfortunately, again, impose a state of tolerance. It is also possible that DCs other than DC ones also do this job. Just for the records, a third mechanism is immune privilege. So essentially that whether the tumor puts or organizes a physical barrier, for instance, by depositing incredibly high amounts of extracellular matrix to physically avoid infiltration from immune cells. So this concludes our first part on the fundamentals. And now we go straight to see how we can use the T cells to kill a tumor. So, and to talk about T cells, actually, before talking about T cell, we have to introduce a main character in T cell biology, which is IL-2. So IL-2 was identified quite early in the history of immunology, or modern immunology, if you wish. And in the 70s, sorry, it was actually christened as T-cell growth factor, which is exactly what it is. So IL-2 is a molecule that is produced by T-cells during priming and also in subsequent phases, but whose primary function is to support, optimize cell expansion, prolong lifespan, and so on and so forth. It is basically a good food for T cells produced by the T cells themselves. It was cloned in the early 80s, and it was used in patients the year after, without any kind of knowledge about the mechanisms by which it worked. So this, of course, gave problems at the beginning, and the first patients were unfortunately a little bit of, recipients of experiments. However, this is how immunology proceeded in the 80s. So the idea of IL-2, if you see it in the context of the cancer immunity cycle, is to act on step 3. So basically, to critically increase the quality of priming of T cells, but not only, also between step 6 and 7, IL-2 is important for T cells in the tumor to optimally deploy their cytotoxic cargo. So it is also a controller of cytotoxicity in the tumor itself. So the first idea ever was to give IL-2 to patients and to increase the immune response against the tumor. And they initially tried with a low dose because they started immediately realizing that IL-2 administered systemically, had toxicities such as flu-like symptom and some kind of hepatic dysfunction. However, those tumor patients had absolutely no benefit. Through mouse studies, it became really clear that actually you have to use IL-2 beyond a certain threshold of concentration. So at high doses, there is a threshold effect. At low doses, it does almost nothing. At high doses, all of a sudden, you start getting something. And this is one of the most clear examples of what you could get. So this is actually 2 metastatic tumor patients, both with liver metastasis. One is renal cell carcinoma. You can see the tumor here in the liver, which basically clears after IL-2. This is a melanoma metastasis patient with a lot of metastasis in the liver, again, cleared up after administration of IL-2. Toxicity of IL-2 at those high doses, a lot, and especially a capillary leak syndrome that was very severe at times. To understand the reason of such a threshold effect to obtain benefit, it became really important to have basic studies about IL-2 biology. And the most important came in 1991, when basically we had available, or it was described, a mouse that was completely knocked out for IL-2. And of course, the first hypothesis about this mouse was that it would have developed immunodeficiency, because IL-2 is so important to support the function of T cells, right? Sure, everybody in shock when the actual result is exactly the opposite. The mouse are not immunodeficient. They have too much immunity. They are actually developing life-threatening autoimmunity. So why is that? It turns out that we had to understand more about the receptor for IL-2. So the receptor for IL-2 comprises 3 chains: the alpha chain, also called CD25, the beta chain, and the gamma chain. When all 3 are present, the receptor is a high affinity receptor for IL-2. Very low IL-2 is able to trigger downstream signaling in cells that have this high affinity receptor. However, who are those cells? Well, it turns out that T regulatory cells are the cells expressing the most CD25. So the most IL-2 receptor alpha. Therefore, they are among the ones to be first triggered when you give low-dose IL-2. This is not the end of the story, though, because all other T cells have a version of the same receptor that lacks IL-2 receptor alpha. There is only receptor beta and the receptor gamma chains. This receptor is a low affinity receptor, and it is triggered when IL-2 is at very high concentration. So which cells do express this kind of IL-2 receptor? Essentially all effector cells, except in a tiny window upon their priming and also activation. So generally speaking, however, the effector cells do usually express the low affinity receptor. So what does it mean? It means that we can support the effector cell, the effector function of T cells when we give a lot of IL-2. This is probably the reason why this biology of the receptor of IL-2 is the reason why we had to give a lot of IL-2 to patients, essentially to see some effects. However, the IL-2 immunotherapy, despite having all the side effects that we discussed, was still used until recently for specific types of tumor, especially clear cell renal carcinoma, which is this KIRC thing here that I boxed in red. And why did it work there? Well, we can only hypothesize, but make hypotheses, but one of them is that those tumors tend to have the highest CD8 cells and cytotoxic cells that are probably a sub class of the CD8 cells, while they have little Th2 and regulatory cells overall. Therefore, the IL-2 in this case is triggering the high the cells like CD8 T cells that may have the low affinity IL-2 receptor. So just to summarize, the history or the current status of systemic IL-2 treatment is as follows. It was interesting at the beginning, albeit it led to severe side effects. However, by knowing more about the biology of IL-2, we understood that we should basically avoid to trigger regulatory cells, which occurs preferentially because Tregs basically express the high affinity receptor of the IL-2 for the IL-2. So current studies are to engineer IL-2s that preferentially bind to the lowest affinity receptor, leaving Tregs out. Another scenario in which IL-2 was actually used was not in vivo, but in vitro to generate T cells that can be infused again to the patient and have a therapeutic effect. Again, we are generating cells that act within the tumor itself at step 6 and 7, and those T cells are endogenous T cells, selected to respond against tumor antigens, or endogenous T cells directly isolated from the tumor. For CAR T cells, which also benefit from IL-2, we'll take a look later in the presentation. Right now, let's review ETCs and TILs. So endogenous T cells are basically derived from a blood draw, and they are stimulated in vitro, with antigens that are neoantigen for that type of tumor. For melanoma, for instance, there is the MART1 neoantigen that is very common. Therefore, by exposing blood T cells to this neoantigen in vitro, in the presence of IL-2, you generate a population of CD8 T cells, in this case, that is MART1, that is reactive towards MART1. And then we give it back to the patient, to melanoma patients. This patient specifically had metastatic melanoma, and had also, after the administration of ETC, a split response, meaning that some of the metastases here went away and some other grew. So why is that? And insight came from immunohistochemistry here. So in the melanoma before treatment, we had expression of several melanoma antigens, including GP100, tyrosinase, and melan A, MART1, which you can see by the brown coloring of those cells. However, after having given MART1 specific cells post-therapy, the melanoma lesions still have a lot of GP100, a lot of tyrosinase, no MART1. Why is it so important? Because it leads us to the mechanism, and this mechanism is actually of general relevance. The point is that the MART1 cells perfectly did their job. They eradicated MART1 expressing melanoma. Unfortunately, though, other melanoma cells were not expressing MART1, and those escaped recognition. This is actually a common problem of many therapies that are too targeted, that are targeted only against one thing. The tumor, in order to escape that therapy, has to do something that is extremely good at doing. They just need to mutate that antigen away. Therefore, hitting only one target is generally a bad idea. Hitting multiple targets, in other words, giving breadth to the anti-tumor immune response, is something that is much more promising, and we are discussing in a few slides. Let's review TILs. Actually, they work very similarly to endogenous T cells. However, they are not derived from the blood, but from the tumor itself. In other words, the selection for tumor antigen may have already occurred since they were tumor-infiltrating lymphocytes. What happened is that this melanoma patient got their melanoma explanted by the surgeon, and then T cells were purified from the tumor and expanded in vitro, no more with not with antigens that were specific for melanoma, but just polyclonal stimulation, again under the assumption that the selection for melanoma reactive T cell has already occurred in the patient, and those cells, the T cells, were in the tumor because they were actually reactive against the tumor. So they just gave polyclonal stimulation, which is a stimulation that basically is good for all T cells in respective of their specificity, plus IL-2. They gave them back to patients, obtaining, in some cases, not in all cases, but in some cases, even complete responses. So this therapy works very nicely, but for selected patients. Well, you may ask, so we are trying to surrogate priming or reactivation of tumor T cells, tumor reactive T cells in vitro. Well, can't we use a vaccination strategy to obtain the priming directly in vivo? Well, yes, that is definitely a great idea, and it was tried by this company, for instance. It was called Dendrian, and set up something like that. So once the patient was admitted to the hospital to get rid of the primary tumor, they also, the physicians also got a blood draw, and they sent this blood to Dendrian. So in this case, sorry, I didn't specify, the primary tumor is prostate tumor. So the blood of the patient was sent to Dendrian. It was processed to basically extract and culture dendritic cells, and those dendritic cells were pulsed with an antigen that is actually predominant, a neoantigen predominant in prostate tumors, which is prostate acid phosphatase. Then these dendritic cells were sent back to the hospital for infusion in the patient. The idea is to basically bypass step 1 and 2 of the cancer immunity cycle go straight to step 3. Those dendritic cells are loaded with a neoantigen for tumor, and they will go to the lymph node, causing a fantastic endogenous T-cell priming. Unfortunately, however, the benefit of this therapy was very limited, with a median survival going from 22 months in control patients to 26 months only. So unfortunately, this therapy gave so little benefit that the company that was producing that filed for bankruptcy. Quite interestingly, the therapy, I think it is still picked up by some other company and still commercialized, because it is, unfortunately for prostate patients, is still something, that kind of works. However, there is a long way to go. And also in this case, the benefit was really limited by using a therapy that was a little bit too targeted. This is indeed the reason for failure. And let me explain why again, reiterate the concept just in this slide. If the therapy is too targeted, the tumor may just develop variants that don't have that antigen anymore, or they don't present antigens at all. In that case, the tumor becomes invisible to the immune system and grows out, killing the patient. This is called also tumor immunoediting. You may find this terminology in some of your studies. However, the problem is targeting too precisely is a problem. Having a breadth of response is much better. And this is how actually the vaccine field is going right now, not to vaccinate against one neoantigen, but 5, 10 of them at once to ensure sufficient breadth. However, stopping to talk about vaccines, we now get to the most important part of today's talk, which is looking at checkpoint blockade immunotherapy. It very nicely connects with the need of more breadth, because checkpoint blockade immunotherapy unleashes several clones of T cells against the tumor, ensuring breadth. And probably this is one of the reasons why it has been extremely efficacious. So just to refresh you how it works, we said a few slides ago that T cells that constantly see their antigens start to develop brakes for further activation. They start to self-inactivate themselves, or to be tolerized, in other words. Those brakes are molecular brakes, and two of them are called PD-1 and CTLA-4. A cell that does not have those brakes is able to more efficiently respond to the tumor. So the idea is simply to give antibodies to remove those brakes and see what happens. And what we think it happens from the point of view of the tumor immunity cycle is that the priming is enhanced, but not only that, also the cells functioning in the tumor is enhanced. This was a good idea based on mouse studies in early years of the century, so early 2000s. And it was tried in people around 2005 to 2010. So to illustrate you why this therapy took the field of oncology by storm, I have to tell you the story of patients with advanced melanoma. This is not patients that have, the melanoma that the dermatologist spots as a little dot on the skin and takes it away immediately. The melanoma has not spread and everything is fine. No, those patients have a more advanced disease. Melanoma has metastasized. And unfortunately, when it metastasizes, melanoma is a very bad guy. So not a huge amount of time ago, just 10 years ago, you could have, or investigators could have, a major paper on the best journal in the world, the New England Journal of Medicine, the best medicine journal in the world, using or showing that they have a new chemotherapy that could prolong the survival of melanoma, advanced melanoma patients of 3 months, essentially. Note that in 2012, essentially everybody diagnosed with metastasis from melanoma was gone in less than 1 year. This is how terrible was the prognosis at that time. And then immunocheckpoint came to age. And how did this picture change? Well, everybody jumped on the chair when we saw not only better response, but durable responses over time. Some of those patients were basically without, they were not experiences a comeback of the disease for 5 years, they are almost cured from the disease, which is amazing for something that just 10 years ago was a death sentence. No wonder that the people discovering this therapy, Jim Ellison and Takuzo Honjo, got the Nobel Prize in 2018. To understand how CTLA-4 blockade therapy works, we have to first review how T cells are activated. They are activated through 3 signals. Signal 1 here is mediated by the T cell receptor. Signal 2 is the co-stimulation, so-called co-stimulation, carried out by CD28, binding to B7 molecules on APC. And signal 3 is mediated by cytokines. CTLA-4 modulates signal 2. This is important. It is modulator of signal 2. And how so? CTLA-4 is completely actually an homologue of CD28 and binds to the very same ligands, to B7 molecules. However, it binds much better. It binds much better and basically sequesters B7 molecules from the availability to bind CD28. In many textbooks, include the Janeway textbook, CTLA-4 is also told or supposed to mediate an inhibitory signal. So basically to transcribe inhibitory genes from the genome. Let me tell you, this is a misconception, because CTLA-4 does not have intracellular domains to trigger the cascade of events that brings to transcription. So, therefore, CTLA-4 works by simply sequestering B7 from binding to CD28. In other words, it interrupts signal 2. When we give an antibody against CTLA-4, though, the signal 2 is restored, and therefore the cells experience a much, much, much better activation. You may have, however, noticed that the majority of patients receiving CTLA-4 blockade do progress. They don't have a long-term benefit. How so? This is actually an intense area of investigation, including in my lab. And we came out recently with a possible explanation on why CTLA-4 often fails. The thing is that T cells, effector T cells, are not the only one expressing CD28 or having benefit from CD28 co-stimulation. T regulatory cells that are immunosuppressive also have a lot of CD28 molecules on the surface. Therefore, they can be enhanced, functionally enhanced, or numerically enhanced by the binding or the neutralization of CTLA-4 through an antibody. And this is what actually Antonio Ribas, one of the leaders in the field, reported in patients. So this is blood from, actually not even blood, I think it is tumor section from melanoma patients treated with an irrelevant antibody or with an antibody, two different ones actually here, that blocks CTLA-4. In both cases, the T regulatory cell density in tumors goes up. We published a paper last year looking at the same phenomenon in mice and asking ourselves whether not the depletion of T regulatory cells, but their inactivation that we can achieve using genetic tricks in mice would synergize with CTLA-4. And this is the result of the experiment. This is colon carcinoma in mice, not treated, all the mice die, treated with a blocking antibody for CTLA-4, and essentially you have no benefit. You deactivate the many Tregs in those tumors and you have essentially no benefit. You put CTLA-4 blockade and Treg deactivation together and you start having a very nice benefit, which again gets us to the concept that tumor checkpoint immunotherapy targets cells that you shouldn't target to have the maximal effect to protect a patient or to kill a tumor. So therefore, in the future, we may ameliorate the outcome of immunotherapy by depleting, by inhibiting the immunosuppressive mechanisms that it also unleashes. So this is the end for CTLA-4. No, it isn't. Sorry. So in this slide, I just want to give you an idea on why the immunotherapy with CTLA-4 works for some patients, but not others. So you all have heard of SNPs, single nucleotide polymorphisms. Those are basically allelic variants that a good part of the population has, and not necessarily they are linked to any kind of genetic defects. They may be just, you know, simple variants like, I don't know, blue eyes and brown eyes. So some people, however, relevant for our discussion now, some people have a SNP in a gene called CD16A that makes the antibody, and especially the antibody against CTLA-4, much more powerful. It turns it from a pure blocker of CTLA-4 to an antibody that also depletes the cells that express CTLA-4. Most of them are, no surprise, T regulatory cells. So in some of the patients, those that have this mutation in CD16A, the CTLA-4 antibody ipilimumab is turned into a Treg depleter as well. And how do those patients do? Extremely better. So it is actually very well possible that part, but not all, of the function of CTLA-4 antibodies is due to some Treg depletion in patients that are so lucky to have this polymorphism in the CD16A gene, that, by the way, is the receptor for those antibodies, is the Fc gamma receptor for those antibodies, which actually is, of course, the bridge between the binding of the antibody and the effect that the antibody has on the cells. In this case, turning the cells it binds to killers of T regulatory cells. So we move to PD-1 now. To understand how PD-1 immunotherapy works, we have again to take a look to T-cell activation. And as you recall from a couple of slides back, T-cells are optimally activated when the T-cell receptor signals and recognizes the peptide bound on his MHC, and when you have co-stimulation using through CD28 that binds to B7. So PD-1, actually, by binding to its ligand PDL1 and also PDL1 and 2, sorry, also PDL2, it basically brings a phosphatase. It recruits a phosphatase, SHP2, and also many other phosphatases that they localize very close to the TCR and the CD28 on the cytoplasmic portion of T cells. And this phosphatase basically interrupts the phosphate cascade that mediates the signaling of the TCR and CD28. Therefore, the blockade of PD-1 interaction with PD-L1 through antibodies enhances T effector cell function by taking away a phosphatase that breaks TCR and CD28 signaling. In other words, PD-1 increases both the signal through the TCR, signal 1, PD-1 inhibition, of course, increases not only the signal through TCR, the signal 1, but also the signal from the CD28, which is signal 2. And it is incredibly efficacious against a very, very huge array, I would say, a huge array of tumors. However, those tumors that respond to PD-1 have a characteristic that is actually found in this graph here on the right. The tumor that respond the most, which is the objective response rate is here on the y-axis, are those that are the most mutated. Most mutation per tumors, like in melanoma, in other skin tumors, or in tumors that are genetically unstable, thus prone to accumulate mutation. And in all these tumors, the PD-1 works really, really great, and almost half of the patients experience a response to therapy. In tumors that are really not that mutated, like, for instance, pancreatic cancer or glioblastoma of the brain, and so on and so forth, the objective response rate goes down quite a lot. Therefore, key concept, the efficacy of PD-1 is intimately linked to how immunogenic a tumor is, to how able the tumor is to elicit a T-cell response. However, there's also need to improve PD-1 because in the best case scenario, melanoma is among the tumor most responsive to it, to immunotherapy. In the best case scenario, you get 40% of response. Now more current data, they probably set this threshold to 50% of the patients. But the problem is that still half of the patient, or more than half, has no benefit. Again, why? And again, here, it becomes important to study immunosuppressive mechanisms that are elicited by the PD-1. Again, my lab is an immunosuppression lab. We study T regulatory cells. We have seen that the T regulatory cells associated to tumor express gobs of PD-1. And when we block PD-1 using an antibody, we expand the Tregs locally. And curiously, the T regulatory cells, they are not the only one to expand. CD8 and CD4s also do, but they are the ones that by fold increase expand the most. We have now data showing that if we prevent this T regulatory cells accumulation, we basically improve a lot the outcome of PD-1 immunotherapy. The mechanism is, by the way, completely different from what we found for CTLA-4. I'm happy to tell you more if you are interested, just email me. So, are those amazing advances coming for free? No, they aren't. We have to pay a price. And the price that we pay is that making the immune system to recognize and fight tumors better, it unfortunately also, paves the way for the immune system to react against things that should not be reacting against, especially healthy tissues. So many of the patients that undergo checkpoint immune blockade immune checkpoint blockade, sorry, experience immune-related adverse events, or IRIEs. And the many systems are involved. In many instances, those autoimmunity events are actually manageable. However, if they involve especially the muscle, they can be fatal, because there is actually description, luckily it is quite rare, but in some patients, the muscle that is attacked is the heart. So the patients develop a myocarditis that can be fatal. So therefore, a lot of the current research on the immune checkpoint blockade is how to prevent and manage this autoimmunity that ensues. So this actually concludes our discussion about T cells. So let's quickly move towards the end of today's lesson by reviewing other cell types. And the first one is B cells that do antibodies. Antibodies are an effector mechanism tried directly to kill tumor cells. Therefore, they are, conceptualized as in step 7, being in step 7 of the tumor immunity cycle. And how do they work? The most important concept is that the antibody recognized antigens in their native form, the proteins as they came out of the cell. However, you may remember that this is in stark contrast with what T cells do, as they recognize proteins that have been heavily processed by the cells. They have been chopped up and loaded on MHC. So they're heavily processed proteins. The characteristic of antibodies is to bind and neutralize proteins that are not processed. And this can be extremely advantageous in a variety of settings. Also because when the antibody binds to the non-processed protein through and it binds through the variable regions here, the constant region, which is the tail of the antibody, so this part down here, is free to bind to effector mechanisms, or to bridge towards effector mechanism. For instance, it may recruit NK cells or macrophages to mediate the death of the cell, or we can actually engineer the antibody to basically bring a toxin to the cell it binds to, or a radionuclide to the cells it binds to, therefore turning antibodies into weapons to get rid of the cells that express that protein. And for some tumors, essentially, for example, non-Hodgkin lymphoma, antibody against CD20 that is expressed by the B cells, and it's called rituximab, is absolutely a godsend, with an amelioration of progression-free and overall survival of going from less than 50% to almost 80%. However, it is a form of targeted therapy with all the problems of extremely targeted therapies that we have discussed before, essentially escape of some tumor variants. And it targets antigens that may be expressed by non-tumoral cells. So here, CD20 is, for instance, expressed by all B cells. So the patients that receive this drug will not only have their lymphoma wiped out, which is great, but also normal B cells, with, of course, the side effect of leading to leaving the patient more exposed to infections and various types of infections, because simply the patients have no more B cells. This is a list of several targets against which therapeutic antibodies have been developed. Don't worry, it is not part of the exam, it is just for your culture. And now that I have introduced to you the antibodies, it is time to talk about CAR-T. So CAR T cells is essentially engineered T cells that recognize the antigens not like a T cell, but like a B cell, like an antibody. And how does this happen? People have engineered a CAR receptor, chimeric antigen receptor, CAR, to be like that. The outside portion, the portion that is basically facing outwards, outside the cell, is a portion of a B cell antibody, B cell receptor that is exactly an antibody. Is a portion of an antibody, thus, and that recognizes proteins in their native conformation. Then there is a hinge transmembrane region, and within the membrane, within the cytoplasm, we find several motifs that actually pertain to the TCR, the CD28, and some other costimulatory molecules. So these receptors recognize the protein as a B cell and signal as a T cell. So the idea now is to introduce these receptors in T cells, and we do it via lentivirus, essentially. So we produce lentivirus containing this DNA for these CAR receptors, and we transduce T cells with them, and the T cells will start expressing the CAR. And for this transfection, also IL-2 is used. And then we have a transfer into immunodeficient patients. So for the CARs that recognize CD19, a molecule that is expressed in acute lymphoid leukemia, the results are fantastic. We have 90% complete remission, while the chemo gives you less than 25, 78% 6-month survival. However, this therapy basically is such that the patients get a very angry T cell in the body expressing or secreting tons of inflammatory cytokines. So essentially, all the patients have very severe cytokine release syndrome, and 30% of them basically are admitted to the ICU. So it is a very toxic therapy, unfortunately, although for some tumors, extremely successful. Okay, in the very last part of my talk, I would love to introduce you to innate immunity against tumors and how we are now trying to unleash its power against cancer. So why innate immunity? And why we basically try to use innate immunity? Well, innate immune cells are commonly found to be a major component of the whole cancer. So if you basically take a tumor, you chop it up, and you analyze which kind of immune cells you get in there, you get that the vast majority of them are macrophages, MPH here. It's a classic short for macrophages. There are some neutrophils, there are monocytes, and the lymphocytes are just lymphocytes like T cells are just a little part. The vast majority of immune cells in a cancer, with very few exceptions, is myeloid, is innate. And macrophages are a big, big part of it. So, and this is one of my favorite things, actually, because it gets us to the beginning of the history of immunotherapy. And it is intimately connected to macrophages. So there was a physician in the late 19th century and beginning of 20th century. So his name was William Coley, that basically was interested in helping patients with sarcomas. And he noticed, after a lot of failures, that patients that not only had sarcoma, but also skin infection called erysipelas, that is caused by this bacterium here, Streptococcus pyogenes, they did much better in managing the tumor. In some instances, they had a regression of the tumor. So what Coley did was actually to culture those bacteria, make a concoction with those bacteria and something else. Those were called Coley's toxin, and basically inject those directly intratumorally into patients. Well, this is extremely crude. However, Coley had incredible results, especially for the time. In this article from 1908, it is claimed that 150 cures out of 430 cases, which is incredible numbers. What is actually true is that this worked in some patients. Some of them actually treated later than 1908, because Coley's toxin were administered to probably the 50s. Some of those patients are alive today as a living testament, so to speak, of this work. However, many of the patients experience side effects from having a bug essentially injected into their tumor. Some of them developed fever, some of them died for the infection, not from the cancer. So it was really something to be perfected. But the way was open, and now we know that what the bacteria were actually unleashing is much likely a macrophage-based immune response against the sarcoma. So as I told you at the beginning of the talk, macrophages are broadly divided into pro-inflammatory M1, those are the anti-tumor MACs. And some immune cells, like dendritic cells, are also able to retromigrate to the lymph node to start the priming or to coordinate the priming of T cells. So much likely, Coley's toxins worked by making the macrophages pro-inflammatory in the tumor. And this concept has been, maintained and developed during the 20th century. However, in an attempt of making the therapy safer, the scientists decided to isolate the components of bacteria that were actually helping against tumor. And nowadays we have several drugs that were developed with that with that concept in mind. One of them is actually you find over the counter right now. It's called Aldara. It's imiquimod. It's basically an agonist of TLR7 and 8. I think mainly 7, and which is the same. The same target that some bacteria actually trigger. So this imiquimod is particularly efficacious against a skin tumor called basal cell carcinoma, here in 2 different histological variants, but you get a complete response of almost 80% of the patients. For even more serious tumors like metastatic melanoma, the treatment with Aldara, with imiquimod, benefits many patients and leads to the regression of many metastases. Unfortunately, in this picture, we can clearly see that even if some metastases or many metastases are gone after inflammation, some are starting to show up again. Unfortunately, demonstrating that the effect of imiquimod is not complete. Similarly to that, we also tried, the physicians also tried to administer just, molecules that are secreted by an inflammatory macrophage, including type 1 interferons like interferon alpha. And the story I wanted to tell you about that is that if you treat with interferon alpha patients with chronic lymphoid leukemia, you actually get a very, very good response to that. And this was tried in the 80s. In this clinical trial, they had 3 complete responses and 4 partial responses. However, there were a lot of side effects by giving interferon alpha systemically, including the worst of which is actually severe depression, suicidal depression in many patients. This is unknown, the reason why interferon alpha treatment leads to depression, severe depression symptoms. And at the very same time, there were also other therapies, chemotherapy with cladribine and pentostatin, which also gave fantastic response. So this drug has been discontinued for now. However, it tells you that taking it is further, example of how the manipulation or the leveraging of innate immunity, so something that is derived in this case from macrophages, can be of help against tumor. The last slide of this presentation is to give you an idea on how the modern science is actually to leverage those concepts right now. So now the idea is to exploit the characteristic of macrophages to be extremely plastic, so you can reprogram those cells, to turn tumor-associated macrophages that are, generally speaking, anti-inflammatory, they are M2, to reprogram them to pro-inflammatory M1 inside of a patient. And this is a frontier. A lot of trials and a lot of studies are being conducted along this reprogramming concept, but we don't have examples of therapy working yet. It is, however, coming up. And I won't be surprised if in a few years from now, innate immunity, innate immunotherapy, we should call it, will become actually a weapon in the arsenal of clinicians. And this is the end of my presentation. I hope it was good and clear for all of you. Since it is recorded, it may go back and forth as many times as you wish. And of course, if you have any question, please email me f.marangoni@uci.edu, and I will try to get back to you.